PCR to test for the presence of the GPCR6 gene in wild-type Tetrahymena thermophila, and the lack thereof in the GPCR6 knockouts as created and described by Lampert et al. Alternatively, we also used PCR to test for the Neo3 binding cassette, which was used in homologous recombination by Lampert et al. To disrupt the GPCR6 gene in the GPCR6 knockout variety of Tetrahymena thermophila.
The GPCR6 gene codes for a G protein in the rhodopsin family used to detect lysophosphatidic acid and proteose peptone. These substances are chemoattractive for Tetrahymena, that is, they swim towards these substances when detected by the GPCR6.
PCR works by amplifying a section of DNA that could then be viewed by running the amplified DNA on a gel supplied with a voltage current. DNA is denatured by heating, and then the section of interest is bracketed by smaller complementary sections of DNA called primers. Thermostable DNA polymerase from the organism Thermus aquaticus can then amplify the section of DNA bracketed by the primers multiple times in order to produce enough DNA to read on a gel.
For our PCR reaction setup, we needed pipettes, an ultracentrifuge, 1.5 milliliter conical tubes, PCR tubes, upstream and downstream primers, instagene matrix at 2 times concentration, GoTac hot start, sterile water, a thermal cycler, a hot water bath, an Instagel, a voltage generator, and of course, wild-type and GPCR6 knockout tetra.
We spun down 1.5 milliliters of Tetrahymena thermophila in a 1.5 milliliter test tube in a centrifuge at 13,000 RPM to separate the Tetrahymena thermophila from their media. We then poured off the media solution and resuspended the Tetrahymena thermophila in 50 microliters of sterile water. In a separate 1.5 milliliter test tube, 500 microliters of instagene matrix was added with 50 microliters of the resuspended Tetrahymena thermophila. The beads in the instagene matrix can settle if left alone for too long, so we made sure to shake the instagene matrix well to resuspend the beads. In order to lyse the cells, the 1.5 milliliter test tube containing the 500 microliters of instagene matrix and 50 microliters of Tetrahymena thermophila was placed in a warm water bath at about 95 degrees Celsius for approximately 5 minutes. After the Tetrahymena thermophila sat in the bath for about 5 minutes, we spun down the test tube again at 13,000 RPM for approximately 5 minutes. From there, we collected the supernatant to use as our DNA template in the PCR reaction and discarded the remaining pellet.
We had 4 experimental setups for our PCR reaction: wild type with GPCR6 primers, wild type with Neo3 primers, GPCR6 knockouts with GPCR6 primers, and GPCR6 knockouts with Neo3 primers. Each PCR tube had a total of 25 microliters of solution, and in each one we mixed together 12.5 microliters of 2 times concentrated GoTac Hot Start Master Mix, 1 microliter of upstream primer for a final concentration of 10 micromolar, 1 microliter of downstream primer for a final concentration of 10 micromolar, 1 micromolar of DNA template, and 9.5 microliters of nucleus-free water. We then placed the well-mixed PCR tubes into the thermal cycler with the following settings: 94 degrees Celsius for 2 minutes, and then 94 degrees Celsius, 58 degrees Celsius, and 72 degrees Celsius, each for 1 minute but repeated 30 times. And we finished up with 72 degrees Celsius for 5 minutes. Afterwards, we held the reaction at 4 degrees Celsius continuously until we were ready to visualize the DNA on a gel.
After the DNA had been amplified in the thermocycler for the appropriate time, we loaded each well with a different sample, and one well with a base pair ladder for comparison. The gel was then hooked up to the voltage current for the appropriate amount of time and afterwards visualized.
The PCR gel we obtained is shown here. Lanes 1 and 2 contained a wild-type DNA template, whereas lanes 3 and 4 contained a GPCR6 knockout template. The final lane contains the DNA template ladder. Lanes 1 and 3 were PCR'd for the GPR6 gene, and lanes 2 and 4 were PCR'd for the Neo3 binding cassette. A band of approximately 300 base pairs appeared for the GPCR6 gene on the wild-type DNA template, and no band for the Neo3 gene showed up. The GPCR6 knockout DNA templates had no band appear for the GPCR6 gene, and a band of greater than 1,000 base pairs appeared for the Neo3 binding cassette with flanking region. This indicates that the GPCR6 knockouts had indeed had their GPCR6 gene knocked out and replaced by the Neo3 binding cassette. The bands had base pairs similar to what this figure from Lampert et al. Showed. We used the same primers as Lampert et al. For the GPCR6 region, and the same primers for the Neo3 binding cassette and upstream flanking region.
Our findings were concurrent with those of Lampert et al. Their gel is shown here alongside our gel for comparison. We obtained these same bands in the wild type, and these same bands in the GPCR6 knockouts. Their gel also showed the GPR6 gene still being present in the GPCR6 knockouts. The gene, however, is contained in the Tetrahymena micronucleus and is not expressed. The fact that we did not find the GPR6 gene at all in the PCR product of the GPR6 knockout only further strengthens the argument that the GPR6 gene is indeed knocked out.
Having a GPCR6 knockout allows us to determine the importance and function of this particular cell surface receptor. Behavioral differences as well as intracellular assays allow us to determine what function the GPCR6 serves when we have a knockout model.
GPCRs are important molecular tools for chemosensation not only in ciliates like Tetrahymena, but also in humans and other animals. Nearly all eukaryotes utilize similar pathways, and the GPCR pathway is no exception.
By understanding the function of this GPCR, we can understand similar pathways and molecules in humans that function in important chemosensation.